All right.
Awesome.
Okay, so this is meant to be relatively informal, and you guys should interrupt me if there's something that I say that doesn't make a ton of sense. So I will just kind of go through how I ended up studying Toxoplasma gondii in the brain, why I think it's interesting. My name is Anita Koshy. I'm both a neurologist here and run a lab that studies this parasite and how it interacts with the brain. Okay, oh, sorry. Okay, Toxoplasma gondii. So this is what it looks like in an EM. So it's a unicellular parasite. So, and it's a eukaryote. So there's a nucleus and it's an obligate intracellular. So in its either the stages we're going to talk about, it has to be in the cell. Generally, it has its own motility to get between cells if it needs to, but it won't survive extracellularly very well. And so the things to notice that I want you just to remember, because they'll come up again soon, is that Toxoplasma has all the normal eukaryotic ER, Golgi, things like that, mitochondria, but it also has these unusual organelles. And hopefully you can see my arrow. Please holler if you cannot. You can see this, which is called the rope trees. And then these things, these dark things called here, which are called the dense granules, literally named because they were dense on EM. And the rope trees are named because they were sort of club-like. So these will become important in a moment. So Toxoplasma gondii, like most parasites, has a sexual stage that's not shown on this slide. And that's because unlike most parasites, Toxoplasma doesn't actually need to go through its sexual stage to propagate. So most parasites have to go through their sexual host and their asexual host to complete the circle. Toxoplasma has this unusual capability to continue propagating in its asexual stage. And the way it does that is it goes between these two states generally, the bradyzoite, which is in tissue cyst, which we'll talk about in a minute, and then the tachyzoite. And so usually people or animals, so Toxo has this very broad intermediate host range. So anything warm-blooded, birds to humans. And that's a huge advantage for us because rodents are natural hosts. So it means when we infect a mouse, that's a natural host, which makes it a very nice model for us. But most animals ingest their toxoplasma, so they'll either ingest it as a tissue cyst and contaminated food, or the sexual cycle, they'll ingest it as an oocyst, which contaminates water if you don't have good, clean drinking water. And essentially, this bradyzoite or the oocyst will flip into the tachyzoite stage, which is just a fast-growing stage that just replicates, gets out of the cell, and then goes to infect other cells. This stage, the tachyzoite stage, will basically disseminate throughout the body of the host. The immune response is specifically targeted at the tachyzoite stage and will clear out tachyzoites from most places, except in these tissues where the parasite is able to flip back into the bradyzoite stage. And the places those are found clinically are the cardiac and skeletal muscle and the brain. And those are really the big places that people have identified cysts. And that ability to encysst allows the parasite to hide, we think, from the immune response and then to cause this long-term infection. And so the CNS is the most common site of symptomatic disease. I think there's some data that suggests it's also the most common site of persistence. You can get persistence in the heart and you can get persistence in the skeletal muscle, but I think the amount of parasites you get to persist are higher in the CNS. That's definitely true for mice. I think there's some data to suggest that's true for humans. So this is a normal head CT. And I just pulled it off the internet, but I show you this to show you, compare it to abnormals. So about 10 to 30% of the US is infected with Toxoplasma, at least based on serologies. And most of those people are going to be just fine. Their head CT looks like this. I can't tell the difference between if you have Toxoplasma or you don't have Toxoplasma, but we think once you have it, once you're seropositive, you're going to have it for a long time. So if you get your Toxoplasma as an infant, so in specific a fetus, then you can have quite devastating outcomes. So this is going to be a first trimester fetus that had congenital toxoplasmosis, and I can just say that because of all the abnormalities. So this brain should look much more like this, and these calcifications are sort of classic, as are the big ventricles, because toxoplasma causes a very specific defect in CSF outflow, and it's the torch organism that does that. I think the other ones do not cause that aqueductal stenosis or these ventricular abnormalities. This is an AIDS patient I saw as an infectious disease fellow. And so again, this brain should look like that brain. This brain has been given contrast. This person was given contrast. And so these areas where you see bright are areas where there's inflammation. And in particular, these things are basically pockets of parasites that the immune response is trying to wall off. But it can't wall it off perfectly because they don't have T cells. So this person had a CD4 count of 4. And for Toxoplasma and many other opportunistic infections, especially intercellular infections, you need your T cells to help control that infection. And then finally, this is chorioretinitis, again, pulled from the internet. And that's just to remind people that the retina is an outpouching of the brain, right? So developmentally, it is part of the CNS. And this is actually the most common manifestation of Toxoplasma across the board and in immunocompetent people. So the estimate is about, I can't remember the number of people in the US, I think it's 20,000 who every year turn up with a symptomatic lesion of Toxoplasma that they've acquired recently. It's about 2% in the US. In certain parts of the world, it's higher, that we think is because of different strains of Toxoplasma have different sort of virulence. Okay, so that being said, the reason I think toxo is quite interesting is because if you're asymptomatic, you have this parasite that persists in the brain and you look fine. There are not a lot of microbes that persist in the brain without causing problems, right? And so that tells you that Toxoplasma has figured out how to either evade the immune response, work with the immune response. Something's happened that allows this persistence, asymptomatic persistence, to occur. And then, of course, from the symptomatic end, we can treat your fast-growing stage of Toxoplasma. There are no drugs that treat the persistent stage. And so from that standpoint, neither your immune response nor drugs we give you will cure you of Toxoplasma. And so, of course, we would love to be able to take every HIV positive patient who is Toxoplasma positive, give them some magical drug and say, hey, you're not going to be at risk to have reactivation of your Toxoplasma, right? So understanding the parasite CNS interaction will be useful from that end. Okay, CNS basics. You guys, anyone who's in neuroscience will know this, but I'm just going to go through this for anyone who's not in neuroscience. So this is what the brain looks like, and the major cells of the brain are the neurons, which are usually in these areas called gray matter, and they've taken a little slice of gray matter there. And they're the big cells that conduct electricity. The oligodendrocytes are the insulating cells that produce myelin. We basically will not talk about the oligodendrocytes. The microglia are the innate macrophages of the brain. They are immune cells that have come into the brain, right? There's used to be big controversies about that, but it's very clear they are not derived from the brain. They just come in very early. And then astrocytes, which were originally thought of as support cells for the brain, for neurons in particular, but are now known to mediate a ton of things like synapse production and blood vessels and the innate immune response. Okay, so Toxoplasma, what do we know about Toxoplasma, or what did we know when I came into the field about Toxoplasma and cells of the CNS? So we knew in vivo, and this is in mice primarily, that cysts resided within cells and primarily within neurons. And so David Ferguson had done this with EM, and Sandra Hallinen's lab had done this with confocal microscopy. And we know there were a couple studies that said that astrocytes, neurons, microglia are equally invaded by Toxoplasma. So Toxoplasma brings its own receptor. So it will stick that receptor into a cell and invade through it. So it can invade any nucleated cell. It does not need a specific host cell receptor. So it brings its own receptor, essentially. It just can go in. And it's known to be very efficient at basically every cell. And then in primary rat neurons, there was one study that didn't really quantify things, but this became sort of the lore. Primary rat neurons, there was induction of spontaneous differentiation, meaning that you'd go from that tachyzoite stage to that bradyzoite stage without manipulating the cell in any other way. And there are lots of ways to make Toxoplasma go from that tachyzoite to the bradyzoite in vitro by adding certain stresses. But this model didn't require any stress. You just put the parasites in and they would differentiate. And then interferon gamma stimulated microglia and astrocytes had been shown to clear parasites, while interferon gamma stimulated neurons did not. And these were done by 2 different labs, but just in the same year. And we'll come back to that later. Okay, so the model of CNS toxoplasma was that clearly not done by BioRender. These are my own poor drawings. The parasite would come into the brain. It would infect an astrocyte, a microglia, a neuron, and the microglia and the astrocyte would clear the parasite. The parasite would turn into a cyst, this bradyzoid and cystic stage in the neuron, and that's how they would persist, right? So the sort of concept was that there were few neurons, but that were infected, but when they got infected, they were going to remain persistently infected. And so what I really wanted to do is be able to, so I started working on toxo just as then microarrays had come up and people had started to show that in a dish, Toxoplasma really modified this host of whatever cells, not neurons. They'd done it with fibroblasts and macrophages, but that like if you had an infected macrophage versus an uninfected macrophages, those differences were huge. And so I thought, well, the neuron must bring something to this, right? Not just that it can't clear the parasite. If they're going to persist for a long time, the neuron must be providing something to the parasite. And so I really wanted to understand this relationship, and I wanted to understand it in vivo, because I always like starting in vivo, because I think you can chase a lot of things in vitro that ultimately don't have in vivo relevance. So I prefer to work the other way if we can. And so I've drawn this cyst as quite close to the nucleus, but more commonly they're out in a dendrite or some sort of process. And so they can be quite far from the nucleus. So I needed a way to be able to identify, hey, this cell is infected with Toxoplasma. And the way I went about doing that is we took advantage of what I was just talking about, the rope trees and the dense granules, but the rope trees in particular, which is that these rope trees, the parasite, when it comes up to invade a cell, it injects in a bunch of these proteins. They're called ropes, smartly. And those ropes go, a number of them, they'll do a bunch of different things, but some of them will affect host cell processes, including ending up in the nucleus. And so the idea was, hey, maybe we can take advantage of these ropes. Much later, we discovered, not we, the field recognized that there was actually a second time when the parasite released effector proteins into the host cell, and that came from these dense granules. And so they're also known as GRAs, and we'll come back to that later. Okay, so armed with the rope tree knowledge, I was fortunate to be in a lab that did a lot of Toxoplasma basic science. Essentially, we put Cre recombinase onto the back of that, one of the rope tree proteins, and then you can use Cre reporter mice. And those are reporter mice that basically only express GFP after Cre-mediated recombination. And then our parasites usually express RFP to make our lives easier to find them. And so it would work like this. See, the parasite comes in, it's going to invade this neuron, it's going to inject in its proteins, which includes Cre recombinase now, and this cell should go green, right? And so the first question was, did it work? And so here it is. This is a mouse that was intraperitoneally infected with parasites 21 days previously, and then this is a brain section, right? And you can see there's a ton of green cells, and we know some of these are neurons. I will tell you, and I'll show you the data for that most of these are neurons, but we know some of these are neurons because of these long processes, right? The only cell in your body that makes long processes like that, or in a mouse body, is neurons. So this was great. It worked. The problem was that in a highly infected brain, you have about 3,000 to 10,000, if you're lucky, cysts, right? Each of these little dots is a cell. And this is a single brain slice, right? So there's 299 or so more brain slices. And so if you just start doing the math, there were way too many green neurons, right? So I spent a lot of time making sure it wasn't a leaky system, but that led us to recognize that this Cree system identified a couple subsets of neurons or any cell, but we think about neurons. So the actively infected one, which is the goal, the ones that cleared parasites, relevant for immune cells, maybe not for neurons. And then the category we hadn't really thought about, but could show ultimately in vitro, was this aborted invasion. So this parasite injects in its rope trees and then doesn't end up invading, right? We could find plenty of evidence for that in vitro once you started looking. So collectively, we call these, when we look at them in a brain slice, toxoplasma injected cells or neurons, TINs. So you'll hear me refer to them because in a brain slice, as I told you, the cyst will often be out here. We can't distinguish who's infected. All we know is, hey, here's a green neuron. And unless we create a whole neuron reconstruction or something, I can't tell you whether that neuron harbors a parasite or does not. And so so this was a little annoying because I had too many green cells. This was a problem if I wanted to go in and at that point do laser capture microdissection to pull out these neuronal nuclei because I really wanted to know about this cell, but I was going to get a whole bunch of these uninfected cells, right? So that was annoying, but it also offered us an opportunity to test this model, right? Because previously we couldn't say who was infected and is no longer infected. But once you have a Cree-mediated recombination event, which is a genetic change, right, that cell is going to express green whether or not it has now cleared the parasite, right? So it allowed us to start to say, okay, well, how does this system work? And one of the reasons to ask that question is just the sheer number of neurons parasites seem to be interacting with. And so we mostly focus, or we did focus on astrocytes and neurons, and that's because for any immune cell, even though I'm showing these as microglia, when you have an infected brain, there's a bunch of immune cells that will infiltrate into that brain, and we don't know if you've turned green. Did you turn green in the periphery and come into the brain, or did you turn green in the brain and eliminate your parasite? So we focused on astrocytes and neurons. And I didn't talk about T cells, but these T cells, which you basically will not see in brain sections if you're uninfected, the toxoplasma infected brain has tons of these because this is the way you or the mouse are going to control your toxoplasma. Okay, so we did a very simple thing. We just defined the lineage of these GFB positive cells, right? So we took brain slices, we stained them for neuronal markers. This is new N for anyone who's interested. So the white arrows mean, hey, this matches up with your new N. This little red arrow I put as maybe it matches up. It does actually match up probably with that neuron, but it didn't match perfectly. So we give it we can't 100% say, gets a red arrow. Here's an astrocyte. So this is GFAP in this brain section. We've also done this using a combination of GFAP and S100B and ALDL1H, the Venn mirror sort of preferential antibody. But that's what that looks like. And then these cells, which don't match either astrocytes or neurons, look like immune cells. And they are, these we only see very early in infection as green, non-neuronal, non-astrocytic cells. And they're generally either macrophages / microglia, which we can't distinguish between, or T cells. We did actually get a couple of GFP positive oligodendrocytes, which I thought was cool, but like only one or two. Okay, so then if we look at this and we just look at astrocytes versus neurons, it didn't matter when we looked, whether it was early, so 10 days post-infection, again, IP infection, or 12 weeks, but it was always neurons that were green. And we spent a lot of time trying to make sure we weren't missing astrocytes that were getting infected, clearing their parasite and dying or something like that. But what we saw was, man, these neurons are being interacted with parasites way more than we thought they were. And so our model, and I think for most people, the model has shifted for toxo of saying, okay, it's not the toxins coming into the brain and infecting everyone. It is probably infecting some cells that we're missing, but it is also interacting with a ton of neurons, and way more than we expected. And then in some number of them, it ends up being able to persist. And so that was a pretty big change in the field, to be thinking like, oh, it's manipulating all these neurons and we don't know about it. And so why did this change our view? So it means that toxoplasma primarily interacts with neurons, which means they're not incidentally infected. And the reason that matters is in the world of host parasite interactions, microbes and host cells end up essentially in an arms race, but they also, for chronic infections, end up sorting out, hey, this is the best place for me to establish a chronic infection without me killing the host. Because if you're something that's trying to chronically infect a host, killing the host generally does you no good, right? So it says that there's something nuanced about those interactions, and it manipulates many more neurons than it actively infects. And so we could show this with some of the other rope trees where we know exactly what they do, that the cells that were green but no longer had parasites in them or never had parasites in them could show some of these effects of manipulation. And there are other people who've gone on to do this work and showed the same thing at a much finer level. And so this then leads this question that we'll talk about in just a minute, but how do uninfected tins arise, right? Is it that neurons can clear parasites, or is there really just this massive amount of aborted infection? And why would you do that? If you're a parasite, why would you waste all that energy to inject in these proteins? Okay, and then what's the consequences of this toxoplasma neuron interactions for the neuron and for the parasite? Okay, so do uninfected tins could arise from clearance or increased abortive infection, right? So you could have this is the simplest idea, which is not supposed to happen. Or you could just have that, hey, in the setting of immune interferon gamma, which is the major cytokine that drives protection from toxoplasma, instead of interacting or invading, you just get a bunch more injection, right? We can't distinguish between the two. And we really wanted to, because anytime you present, I would present our work and there was a virologist in the audience, they would say like, yeah, neurons aren't clearing this. You haven't convinced me. This is what's going on. There's just a ton more abortive invasion, because the virology field has a whole history of that. And so we really wanted to say like, okay, do we think clearance is happening? So the first thing we want to show is, does interferon gamma actually show a decrease in infection rate, right? So there's this historical data that said it doesn't. And we were just going to re-ask that question based on, hey, we have all this, all these things that look like uninfected neurons. And so we just interferon gamma stimulated or didn't stimulate neurons, and we asked, what's the rate of infection, right? So we could definitely see a drop in the rate of infection in the setting of interferon gamma stimulation. And the reason we got this answer and the prior people got a different answer was actually the strain type that we used. So there's these different strains in Toxoplasma, and in 2001, everyone thought they weren't that different. They're pretty similar. The strain type known as a type 1 strain, which is what most people use because it's much easier to grow and genetically manipulate and things like that, it turns out that in mice in particular, it has all the virulence factors that defend specifically against mouse mechanisms to destroy Toxoplasma. And so the lab that did the astrocyte work actually used a type 2 strain that doesn't have all the virulence factors. So they saw this drop, and the lab that used the type 1 strain in neurons saw no drop. And we've gone back and done this in this paper with the type 1 strain, and then we get no change. These are mouse neurons. We get no change because it has all the virulence factors, and it just defends itself against what the mouse mechanisms. Okay, so we could say, all right, we now understand why there was this discrepancy in the literature. There is a drop, but it didn't tell us that we had clearance, right? You could equally draw out this rate with these guys have more invasion, these guys have less invasion, things like that. And so we really wanted to say, like, can we prove that clearance is happening? And so we went back to use Toxoplasma biology. So right now, right, we're stuck on the rope treat Cree system. And I told you there's this GRA system, right? And those guys only really get released once the parasite is fully invaded. So now if we stick Cree on the back end of one of these GRAs, right, what we should do is get rid of aborted invasion. So the only types of neurons we should get are actively infected and cleared. Now, so we did that in vitro first, and we could show again, okay, so here's our GFP, that means interactive with cells, and then here's T. gondii, when there's no interferon gamma. Here's an interferon gamma positive culture that's been stimulated with interferon gamma. It's got GFP, but you can see like this doesn't look like this, and we think these are dying parasites in here, this little bit right here. And so, so we really thought we could show clearance, and we could show it on a, again, just counting, right, this GRA-Cree system. Now, what do we see? And again, we see this drop. It's a very similar drop. It's a couple of percentage points off, which actually may mean that sometimes there's an increase in or a decrease in invasion in the setting of interferon gamma. But the majority of this in neurons is driven by clearance, at least in vitro. But of course, you can always, and then we wanted to say, sorry, so these are mouse neurons. We generally treat mouse toxo or we study it, but we would like to know about humans. So we want to know if interferon gamma stimulated human neurons also showed anti-parasite defenses. And so we did the same thing. We took human neuronal cultures, and then we stimulated or didn't stimulate them with interferon gamma, and we quantified the rate of infection. And so again, human interferon gamma stimulated neurons show a much greater degree of decreased infection rate than mouse neurons, which is interesting for a number of reasons, and may explain why mice seem to have a tougher time with toxoplasma than human inbred mice, right? We're always using inbred mice. But we can't say here because we don't have interferon. We don't have all the tricks that we have in mouse neurons that we have in human neurons. We're working on setting up Cree reporter systems in these human neurons. But right now, all we can say is interferon gamma generates a very robust anti-parasitic defense, but we don't know how that defense is mediated. Is it clearance or is it invasion, blocking invasion, some combination between the two? Okay, and then we also wanted to know about in vivo, right? Because it's great to show. I just told you we really like to do things in vivo so that we can say like, hey, it doesn't just happen in dish. So you can do genetic ablations, specifically trying to remove interferon gamma receptors or IRGs, which is the system mice use in neurons. But that's more difficult. I don't know how many of you have worked with Cree neuron drivers. They work great in the Cree reporter system, these pan-neural drivers, but there's lots of literature and in our own experience that when you're trying to remove a specific gene, that rate of removal is much harder. And so, so they're not like T cells or endothelial cells. As always, neurons are very painful. So that wasn't going to work. So you could use parasites to remove the genes, right? We have these Cree parasites to try and do that, but it may be too slow or too late, right? Because when you get an infected brain, you have a ton of cytokine in your brain before the parasite really gets there, right? So if the parasite goes in to try and remove a gene, it may just be too late. The proteins are there. Whether or not that gene is turned off may be irrelevant. And so maybe we could use these G-Crees in our Cree reporters, right? So maybe if we could do whole, since the neurons are big and parasites can be wherever, if we can do whole neuron reconstructions, if we can see a green neuron that doesn't have a parasite in it using the G-Cree, we can be fairly confident that that neuron cleared the parasite. And so that's what we did. And so we being a grad student named Emily Merritt, spent a long time doing these neuron reconstructions. So these are 200 micron thick sections that she would clear and then map. And she would spend a lot of time, and if she thought it was uninfected, she'd spend a lot of time making sure she was mapping in an area where she couldn't see parasites, right? Because she didn't want to say like, hey, this is uninfected, but really it's just that there's this little small break that she's missing in one of the neuron processes. And then if it was infected, that becomes much easier. It's hard to see, but the parasites express DAPI. And so you can confirm that this is a parasite by looking for these little bits of DAPI in there. We ultimately ended up remaking this line to have RFP, and we did it a couple ways. But when Emily did this, what you could find is out of 35 neurons, 17 of them did not harbor any parasites and 18 did. And so it was like a 50% difference. So we could feel quite confident that in fact we can say this happens, right? So neurons can clear parasites in the setting of interferon gamma. Okay. Now there came this other bizarre question that we didn't know quite how to answer, right? So most of the idea about neurons not having immune responses is because many times the way a T cell deals with an infected cell is it kills it, right? So these are called T effector cells that will release things like granzyme B or granzyme A, which are pores which will create pores that destroy the cell, and that's the way you kill the infected cell. And the idea behind neurons not having very good immune responses is that you really don't want your T cells to come in and kill all these neurons, right? It turns out with toxo, the T cells don't generally destroy cells. What they do is they produce interferon gamma that then tells the cell, put up all these defenses, and those internal cell intrinsic defenses are what kill the parasite, right? But so the idea is that you want to preserve these neurons. And so we knew for a long time that if you just counted these green cells, right, green neurons in a brain, at 3 weeks, there are a lot. And by 8 weeks, there were way fewer, right? And that made no sense because in theory, you would want to preserve all these cells, right? So what was going on? And what the dogma would say is that it's general information. So here's a section of a brain. This is a cyst. You can see the green neurons, but all these other little red cells are T cells. And sometimes we'll overuse the same color because we can distinguish parasites pretty easily from T cells, but it just helps us in a 4-color system where we're stuck. So dogma says essentially you have this massive inflammatory infiltrate, and that all the cytokine release and macrophages and things like that essentially end up killing a bunch of these neurons. But it's really hard to prove that, right? Because these things we can count over time. It's really hard to prove that we're missing neurons in very patches. People have tried to do that, and just random patches of the brain, it's hard to prove that you have a lower density when you're toxo-infected. And so we thought, well, maybe electrophysiology can help us. And so if we could patch clamp onto single neurons, right? It's a classical assessment of neuron function and state. And we could compare TINs to bystander neurons. So in ex vivo brain slices infected intraperitoneally, could we compare the ones that were near a green neuron but weren't green? Then maybe we could see if they looked similar, right? If it's just inflammatory milieu, then those neurons should look similarly abnormal. And so it might allow us to account for the effect of inflammation, but it's a non-trivial experiment. And part of that is that it's enabled by the Cre system because you can't patch on to green neurons without the Cre system. And you have to compare apples to apples. And what I mean by that is in the cortex, right? You can have 2 neurons side by side, and they can have, in just an uninfected mouse, they can have totally different physiology because one's a pyramidal neuron and one's an interneuron, right? So we needed a way to say, like, we weren't going to see these big discrepancies just because there was this difference. And so a grad student named Oscar Mendez had mapped these neurons, and he had found that in the basal ganglia, there was this enrichment for these GFP positive neurons. And the reason that's useful is in the dorsal basal ganglia, striatum, 90% of the neurons are a very specific type called medium spiny neurons. So now if he focused on the dorsal and the basal ganglia, he could potentially compare apples to apples. And so that's what he did. So what he did is he used uninfected mouse striatum to prove that he could get normal medium spiny neurons, right? Like he has the right technique. And then he went into an infected mouse and he would patch clamp onto bystanders and onto a tin. And these were not done in the same section, because when you patch clamp onto something, when you pull that electrode out, it creates a hole. So you can't just go, hey, from this cell over 3 cells. Okay, so they were done in different sections, but they were all infected mice. And so this is just to show you the basic idea. So he'd patch clamp here, and then he would go anywhere from 30 microns, so that's 3 cell bodies generally, to 100 microns away to patch clamp on a bystander cell. Again, not in the same section, but general idea. This is the basics of E-physiology, which is not that I want you to know or learn or anything like that, but just to know there's a bunch of different parts you can measure. And this one's going to be important, which is the resting membrane potential. So every neuron type has a very specific resting membrane potential, and you have to go over that resting membrane potential to get it to fire an axon. And that's really what I want you to know. Okay, so if you look at bystander neurons, so these ones are the uninfected neurons, so meaning neurons in an uninfected brain, and then these are bystander neurons, so close to a green neuron, but not green. And if you look at this, they pretty much are the same in all these parameters, except for this resting membrane potential. So the bystanders have a slightly higher resting membrane potential than in the uninfected brain, than a medium spiny neuron in the uninfected brain. All right, so then what about the TINs? So this is just resting membrane potential. Here's the bystanders, here's normal. The TINs, and Oscar was only able to patch clamp onto 10 of them, are much higher, right? So their resting membrane potential is very high. And there are a couple things that can explain this, including, hey, these aren't neurons, right? You patch clamped onto some random cell, not a neuron. So what Oscar did in that scenario is he would bring the resting membrane potential down by shoving in a bunch of electric current, negative electric current. So he could bring this down further, and then he would slowly raise it to see if he could trigger an action potential, because this resting membrane potential is actually above the level of where you would trigger an action potential. And so when he did that, he could start getting, so these are just the different phases of him going up. He shoved in a bunch of, let's see, tins. He then I can't remember, it should say, oh, that's this. I don't know how much current he's put in here, possibly 180 picoamps to get it down. And then he's just going up to try and trigger. And if he did that with tins, he put in a bunch of, so this one is patch clamped, its resting membrane potential is -33. He puts in a bunch of negative current, and then he starts doing this, he can get some action potentials firing, right? So it at least tells you it's a neuron. And in some of them, he could actually get them to act pretty normal, right? So if he put in enough current, got it lower, he could get them up. And so that to us said, these are sick neurons, right? So there's something about interacting with Toxoplasma, or the immune response only causes its damage very, very close to the green neuron, meaning less than 30 microns away, which is not generally how people think of the immune response, but it's possible. And so, so we think there's something about the Toxoplasma interaction that essentially sets off a death cascade. And that's interesting because neurons, again, are not supposed to die very easily. Okay, so summary of future directions: Toxoplasma assists in neurons in part because parasites primarily interact with neurons. We've also shown, and we've done slightly more careful studies, that in fact neurons do at least induce encystment. So that's probably part of why they persist, like way better than a fibroblast or other cells. Yep, there may be neuron-specific interactions. So injection with these proteins alters neurons. Interferon gamma stimulated neurons mount antiparasitic defenses. The murine neuron system uses this IRG. All cells in a mouse use the same IRG system. There's some variants on it, but the basics are about the same. And human neurons, we don't know, because human neurons use a lot more, not human neurons, human cells use a lot of different mechanisms to control T. gondii, and it seems to be cell specific. So different cells use different mechanisms. And so we're in a tool building phase where we'd like to be able to understand exactly what our human neurons doing. Yeah, and we'd also tool building in terms of trying to prove that human neurons are actually clearing parasites. Okay, with that, I will stop. These are all the people in my lab currently. These are 2 undergrads, senior scientist, lab manager. Those 2 are here and here. These are all the guys who helped put together the story I just told you. Most of them are here. So with that, I will take questions, and thanks for your attention.
Thank you. That was super interesting. Um, I have a question right off the bat,
Certainly.
which is a little general, so I apologize. What is known about Toxoplasma gondi's effects on calcium channels?
So not much, meaning, so there's one paper out there where they showed that calcium fluxes in vitro. You could see either more calcium or less calcium in toxo-infected neurons in vitro. But that's pretty much it in terms of calcium flux. When Hannah Johnson, who was an MD, PhD, neuroscience graduate student in my lab, she did some RNA-seq analyses, and what she could find was not that there was a difference so much between neurons in the infected brain or in a dish versus uninfected brain or a dish in calcium channels, but she could find a difference in potassium channels, that they were downregulated in cultures that had been infected with toxoplasma and in neurons in the brain. And that's a slightly complicated study, but there's some evidence that even in vivo, that's also happening.
That's really interesting. I would wonder if maybe that's a feedback mechanism and if there's a way to image the protein receptor interactions on the intracellular side in the tins. I think it would be really interesting to see that.
Yeah, yeah, like we would love to do much more neuroscience inside of like understand, well, what is going on right now? One of the problems for us is the death of the tins and the abnormal, not the abnormal physiology, because we haven't done that, but these things dying is not true in vitro. So even if we dump interferon gamma on the cultures and then infect with neurons, then infect with toxoplasma, what we'll see is the green neurons in that culture will actually look better than the surrounding neurons. And so, like many microbes, Toxoplasma has anti-apoptotic proteins. And so we think that's what's going on, is that the parasite is actually now in the setting of an in vitro setup able to protect the neuron. We don't think that happens in vivo. And so we're trying to figure out how do we do something like you were talking about in vivo, because if we could replicate the phenotype in vitro, that would make our life easier, but we haven't been able to. And obviously part of that is in vitro. They don't tolerate toxoplasma infection for as long as in vivo.
I have a quick question. That was really fascinating. I just have a question about I think you talked about the effector proteins. What are the purpose of those? Like, does that help with persistence? Is it,
Yeah.
to keep toxic just in the
Yeah.
cell? Like, yeah, yeah.
So most of the effector proteins have been studied primarily in the acute phase. And so we know for the tachyzoid, essentially these effector proteins come in and they mess with a bunch of host pathways. So one of them is STAT6, and that only happens with certain strains of Toxoplasma. But if those strains of Toxoplasma don't, and what that effector protein does is it literally goes to STAT6 and phosphorylates it.
Mm.
And that then means that STAT6 will ultimately, as a transcription factor, they'll go to the nucleus and set up this, a bunch of different genes being expressed. And that parasite does that because STAT6 turns down the immune response in immune cells. It means you are more likely to produce less inflammatory and more wound healing macrophages than pro-inflammatory macrophages. And we can show that if you get rid of STAT6 or the effector protein group 16 in that parasite line, that when you put it into a mouse, it actually gets cleared out because it needs that effector protein. And so there's a bunch of them, and they do things like blocking apoptosis, controlling cell cycle, blocking, in some cases, blocking NF-kappa B, in other cases, stimulating NF-kappa B. And the idea behind stimulating NF-kappa B is that, again, if you want to persist, you have to help the host set up the right immune response so that you don't kill the host, nor does the host kill itself with too much of an immune response. And so it's like these really fine interactors, and we probably only know what some of these parasite effectors do because they've been studied in very specific cell types. And at least for STAT6, for example, we know that that phenotype that you see differs between, say, neurons and a macrophage.
So doesn't matter what kind of like toxic strain, they all exhibit the same effector proteins. Because I know you said they're not tropism specific, like it's not one cell type, it's any
Right.
cell type.
They
Okay.
can affect them, and they all, the caveat I would put is for the rope trees, there seem to be some differences between the Toxoplasma strains. And so, for example, that one strain that I told you blocks mouse immune responses, it basically expresses two proteins, the virulent alleles of something called ROP5 and something called ROP18. And because it expresses both, it kills all mice because it just dismantles, like they can't kill the parasite. A very closely related strain is very avirulent. And the reason it's avirulent is because it has an insertion basically in the promoter of ROP18. So it carries the virulent ROP5, but it doesn't express ROP18. And so it gets cleared because it can't defend against this mouse system.
Okay.
So for the GRA proteins, those seem to be those seem to not be strain specific. The ones that come in later seem to be every parasite strain has the same ones. And it's interesting because in vitro, at least, when people have looked at the slightly different timing of when the ropes are released and the GRAs are released, some of the rope free proteins actually set off all these immune responses in the host cell. Like the host cell detects those. It says something bad is going on, and the GRAs come around and then dampen it. So
Okay.
the GRAs are there to be like, yeah, you set off this immune response. Now we got to hide it.
Wow. Oh, thank you.
Sure.
Another,
Hi.
sorry.
Hi. I do have a question as well. Um, when you mentioned doing in vivo or in vitro work, were you transfecting these cells or by some method like that, or were you isolating the cells from the genetically modified mice and then
Yeah.
running your experiments?
Latter. We just take the Cree reporter neurons and then infect them with Toxoplasma, and then
Okay.
add interferon before.
Interesting. Okay. And then, thank you. Sorry. And then Toxo crosses the blood-brain barrier. Have you done, or is that like a common thing that's already been in the field where anyone's looked at it, interactions with the blood-brain barrier?
Yeah, so how Toxo is thought to cross the blood-brain barrier in one of three ways, which are the same ways that viruses are thought to cross the blood-brain barrier. Well, two of them are the same as the way viruses are thought to, because Toxo is modal. There's some in vitro data suggesting it can tunnel between endothelial cells, so it can just go through the ZO1 and all the things that cross-link. So that's one way. I would say that system has the least data, it only has in vitro data. The second two ways are the Trojan horse idea. So toxin infects an immune cell, that immune cell then crosses the blood-brain barrier, because once you have a bunch of inflammation, it sort of licenses immune cells to come into the brain. And then the second way is you infect Toxoplasma, or the third way is Toxoplasma infects an endothelial cell and then ruptures back out the back end of it. So now it's crossed by going through the endothelial cell. Does that make sense?
Yeah, that does. That's fascinating. Thank
Yeah,
you.
it's pretty
That's
interesting.
amazing.
There's some actually really cool papers looking at Toxoplasma and the motility it gives parasite. And one of my colleagues has done in vivo uh, intracranial imaging, and she can watch these T cells that are infected with toxo like cook around the brain. What she hasn't seen is in abundance is the T cell crossing into the brain. So she knows they traffic around the brain, um, but it's a little hard. She can't 100% say, hey, that T cell got infected externally and then came into the brain. So we're waiting for that direct evidence still, but there is other evidence that suggests that's probably the way it goes.
Wow. All right. Thank you so much and fantastic talk. Thank you for your time.
Thank you.
That's crazy.
So,
Really disturbing all around.
also, I'll tell you this. So, because of our work saying like toxo primarily interacts with neurons, there is a group, a non-toxoplasma group in Israel that decided, hey, we should look at using toxo to bring proteins that it'll inject into neurons and all these degenerative disorders. And the reason they were interested is because viruses can only be packed with they have a limited size. And so that Israeli group got in touch with the Toxoplasma researcher who's Israeli, and she got in touch with me, and I was like, that's crazy. Like, who would tell their person to get infected with some transgenic toxoplasma to try and treat this? But ultimately, they did a lot of work on it. And actually, they're the ones who showed which GRA protein could tolerate proteins being attached to it. Um, and I'm on a paper with them because they convinced me that, look, like this is how adenovirus started, right? Is like you didn't understand a bunch of things and then you have to understand it, but it is a reasonable thing to try and think about. So there's a sort of a proof of principle paper out there to say like, hey, maybe we should be thinking about this. Just to be clear, as of now, I think that's insane, but maybe as you de-risk this parasite, it will make sense.
I'm glad we're on the same page about the craziness.
Yeah.
One of the things I was wondering is really what kind of behavioral phenotypes show up in people who have the infections, especially in the persistent phase, and seeing the concentration of where those infected cells were. That seemed
So
concerning.
a couple of things to know, which is that mice, again, B6 mice get many more parasites probably than humans do. And there's a decent amount of data for that from different labs, not ours. But the thing that people, if you look in the literature and you Google toxoplasma, it'll come up as schizophrenia is the most common thing people have associated with it, OCD, and then Alzheimer's and Parkinson's. The thing about those studies, and there's stuff about, toxoplasma people are terrible drivers and they have hypersexualism and all this stuff, is most of those studies don't ask the flip, which is, oh, well, maybe if you engage in high risk behaviors, you're just more likely to get toxo infected, right? Because you eat raw meat or you are willing to drink water that you don't know whether or not it's clean, things like that. And so the way I try to think about it is, so toxoplasma serology across the world ranges probably from 0 potentially, but 10% at a low end, to in some parts of Europe and South America to 60 to 90%, right? So there are places where toxoplasma is quite common. And if it was a driver of, say, schizophrenia, then you should see big differences in the incidence of schizophrenia. And particularly, you can think about like the US and Europe, because those are similar socioeconomic status and cultures. And you don't see a change in those. And so I think of toxoplasma as, hey, if you are predisposed to schizophrenia, do I think toxoplasma can be the factor that tips you over? Yeah, absolutely. But do I think at a population level, Toxoplasma is a big driver of schizophrenia? No. And so I think that's true for most of the things that people try to associate Toxoplasma with.
That makes sense. And actually, that's similar to how I think about cannabis,
Yeah,
with
that makes
people who
sense.
have like psychotic breaks with
Yeah,
cannabis. Yeah.
right. It's just the thing that triggers it, right? It's not.
Do you have any concept yet in the field of what physiological factors might be present that would make someone more susceptible to that, like breaking of the camel's back? You know what I mean?
Yeah, I don't. I think it was I would say no. I would say there are people who work on behavior who I think are trying to get at it. And I think a couple things have come out of the animal studies, which is for a long time, people would say that it was the cyst causing it, like it's something about the cyst changing the neuron. Because there's so few cysts compared to numbers of neurons in the brain, I think that's very unlikely. I think most people have now agreed on some level with that. And the field is more settling on just the inflammatory response, that you get such an inflammatory response. And we know that inflammatory responses are what we think of as um, inflammation, or immune cells actually are important in setting up synapses and destroying synapses. And so you can imagine that if you just happen to get your toxo, you're predisposed to schizophrenia, you happen to get toxoplasma and a lot of it in the basal ganglia, or you set up an immune response, that's going to alter those pathways and specifically synapses in a way that now tips you over. But I think I think there is data for some of that, but it's a long journey before we actually get to the molecular.
That's really interesting. I think it would also be super interesting to see a study where you could get more of a chronological change over the course
Yeah.
of the infection. That would be so amazing to watch.
So it's really easy in mice, and people have done these and have shown, where you see these changes, and you can map them potentially to where parasites are. In humans, obviously, that becomes impossible because we don't have a way to say, like, okay, you have cysts in these locations, right? And we don't have drugs that we can say, like, okay, well, let's get rid of all your cysts, or let's get rid of any inflammation that we don't know about in the brain, right? And then see if that resolves your issues or prevents you from developing issues, right?
Yeah, that's really interesting. Also, Melissa texted her laptop died, and so she
Okay,
wanted me to extend her apologies for rapidly disappearing. And that's fine.
I appreciate it.
Uh, Priscilla and Ingrid, do you guys have any more questions for Dr. Koshy? No? Okay, well, that was really, really cool.
All right, good. Glad
Thank
you
you.
enjoyed it.
Thank you for
Found it
talking to us.
followable.
Yeah, absolutely. It was very logical. And once I post the video up on the website, I'll send it to you as well so that you have access to that.
Cool.
Yeah, thank you.
Thank you, guys. Thank you for inviting me and being such gracious people.
Thank you.
Thank you.
Bye, everyone.
Bye.
Bye.